Okay, are you people able to see the screen?
Yes, ma'am.
All right. Okay. Give me just 2 minutes. I'll start class.
Okay.
Okay, am I audible, students? Yes, everyone, switch on your video. Let's start today's class. Yes, everyone, switch on your videos. Come on, quickly. Let's start today's class. Everyone can hear me, right?
Yes, ma'am.
Okay. All right. So in the previous class, we had discussed about the properties that make the genetic material, right? So let us quickly revise those topics, like the terms or the things that make the genetic material, you can say, acceptable, or what are the eligibility criteria to become a genetic material. The first thing is that any genetic, no matter whatever it is, if the thing is, it is in the line of being a genetic material, then it should be stable in terms of its structure and in terms of its chemical nature. Apart from that, the genetic material, whatever it is, it should be able to make its copy, meaning replication should be possible. Then it should have the scope of slow mutation. Slow mutation means it should allow the, you can say, the introduction of new characters, but at a slow pace so that it is beneficial. Okay, now moving forward. And the most important thing is that the genetic material should definitely be, you can say, able to express itself in the form of Mendelian factors. You people remember the genetics chapter? The way, meaning the genetic material or the part of genetic material that can be expressed in the form of Mendelian character, in the form of genotype and everything. So these are the properties which makes the genetic material. Now, the reason why DNA is preferred as, or you can say preferred as the genetic material over RNA, is because DNA offers all of these properties. Now, talking about why RNA, even though in some organisms, the RNA can also be the genetic material, but why is it not? See, we know that in RNA, the second carbon has an extra OH group, which makes it more labile. Labile means makes it more easily altered. And where is everyone? Why can't I see any faces here? Long time no see. Hmm? Yes, everyone switch on your videos. Come on quickly. Where were you?
I was in the last week.
You were, yeah. Okay. Khadija, are you there in the class? Like, it's just your video icon is showing, your audio icon is not showing. I don't know. Can you leave the class and join again? All right, so talking about the RNA. So the RNA has an extra 2' OH, and because of it, RNA is very reactable and it is very, like, it can be easily altered. So again, it's not stable, and if anything is not stable, then it cannot be considered as a genetic material. Where are you, Mariam? Are you like at your home or somewhere else?
I'm at my home. I'm just in a different room because my dad is
You're at your
working
home
and
only, right? Okay, now you'll be attending the classes regularly or still
yes.
AWOL?
No. A little turbulent.
Okay. And have you seen the recorded lectures?
No.
Okay. All right. Anyway, moving forward, see, since RNA has uracil instead of thymine, so RNA again becomes a little unstable, and the presence of thymine instead of uracil in the case of DNA gives it more stability. Apart from that, the RNA can mutate at a very faster rate because it is unstable, but DNA has slow mutations. So that is why RNA cannot be considered as the genetic material. Now, moving forward, talking about RNA world, the most surprising thing to know is that DNA was not developed first. Like, DNA was not present first. First thing to, you can say, be present on the earth or in the world was RNA, in fact. So RNA was the first genetic material, as many life processes such as metabolism, translation, splicing, and everything, they evolved around RNA. Meaning they happen to go around RNA because they need RNA as one of their, you can say, steps or in the middle one. Now RNA, it used to act as a genetic material, and it used to act like a catalyst as well. So it has dual properties. Now, RNA being a catalyst, it was very reactive and definitely unstable, because catalysts, they are responsible for, you can say, decreasing the rate of, increasing the rate of reaction, right? And how do they do so? Because they are reactive in nature. So RNA was a catalyst, and so it was reactive and it was unstable. So during that course of time, the DNA evolved from RNA. Always remember that. Now, apart from all the above characters that we had discussed about DNA, apart from all of that, the DNA, it has double strands, right? Two strands of DNA are there, and it had complementary strand, meaning one strand is complementary to another strand. So because of these two properties of DNA, it resists any changes. That is why when the DNA gets transferred from one generation to another generation, not a lot of changes come along. Okay, a little bit of changes are there. So RNA world is clear to everyone? I'm waiting for your answers. Yes or no? I can see you. The rest of, yes, clear everyone?
Yes, ma'am.
Hmm. Today we're running low on energy. Why? I I am going to do a petition to keep your classes during the week. I don't like weekends with you all. Like, you people are one of my best batch, and you're like drained out of your energy on Saturdays and Sundays. Friday, you people also get off, isn't it? And what
Yeah.
happens? Like, you get recharge after Fridays, that you people are so dull on Saturdays and Sundays, and you are not there only.
Weekdays are better.
Hmm?
Weekdays are better for bio class.
Weekdays are better, yeah, definitely. All right. Now moving forward, talking about the replication of DNA. Now see, the replication of DNA is known as semi-conservative. Can someone tell me why is it known as semi-conservative? Why is DNA replication semi-conservative? I told you about this thing in the previous class. Who can tell me? Ikra, can you tell me?
Ma'am, can you repeat the question?
The question is, why is DNA replication known as semi-conservative? See, since if I say that this is the DNA molecule, okay? Now, when DNA replication takes place, this twisty topsy thing is there, it opens up. So it sort of looks like this. Okay. Now, each of these two openings, they synthesize one strand of themselves. So when we talk about the new DNA, so each of the DNA has one new strand and one old strand. Because of this very reason, the DNA replication is known as semi-conservative. Okay. Now, while giving the DNA double helical structure, Watson and Crick also proposed the replication of DNA. We know that the modular structure or DNA double helix model, it was given by Watson and Crick. So at that point of time, since they studied the structure of DNA, they also explained to us about the replication of DNA. So they said that DNA replication happens and it is semi-conservative in nature. But again, it's not enough. Just saying is not enough, right? So the proof of it was also done. So what happened? The proof of semi-conservative nature of DNA replication, this proof was given by Matthew Meselson and Franklin Stahl. Meselson and Stahl in the year 1985. So what they did is that they basically grew two DNA, one with N normal nitrogen and one with N14. And when they replicate, like the DNA replication was completed, then what happened? The DNA which was there, it contained one strand of N15 and one strand of N14. Again, one old and one new. Because of this very reason, they said that the DNA replication is semi-conservative in nature. Okay. So basically in this particular thing, when the DNA was grown, or you can say the nitrogen 15 DNA was replicated. So it contained nitrogen 14 and nitrogen 15 as well. Clear? So this was semi-conservative. Now, Taylor and his colleagues, they did the same exact experiment and came to the same result. But the you can say the subject of their experiment was different. It was, they did it on a plant, which was Vicia faba, that is faba beans. And they did it in 1958. Now moving on to the different types of RNA that are present. We have to study about each of these RNAs. So mRNA is there, rRNA is there, and tRNA. The M stands for messenger, so messenger RNA. The R stands for ribosomal, that is rRNA. And then the T stands for transfer, that is tRNA. So the first one that we're going to talk about is the mRNA. Now, remember that in the cell, all of these different types of RNAs are present, but the only thing is that their concentrations are at different level. For example, if we talk about the mRNA, so 5% of total RNA in the cell is mRNA, only 5%. mRNA is the longest RNA and it is also called as a template RNA, nuclear RNA, or informational RNA. So these are its synonyms. Now, what does it do? It carries the genetic information that is provided by the DNA. Now, students, do you remember central dogma of molecular biology? Does anyone remember central dogma? No one remembers. Do you remember that the information flows from DNA to RNA to protein, right? So from DNA to RNA, the information or the genetic information is carried by the RNA, right? And this RNA is actually the mRNA in the central dogma. So this is what I mean by saying that it carries the genetic information which is provided by the DNA. Now moving forward, talking about the rRNA. So 80% of the total RNA in the cell is ribosomal RNA. Now this ribosomal RNA, it is smaller than mRNA in size and longer or larger than tRNA. Now what does it do? It is responsible for performing structural and catalytic role during translation. Translation is protein synthesis or protein formation. So during the protein formation, the rRNA does the process of, like you can say, it provides catalytic role. Catalytic means it does the process of catalysis or works like an enzyme, and it also provides a structural role. Now talking about tRNA. Now tRNA comprises of 50% of the total RNA in the cell, and this is the smallest RNA. It is also known as adapter RNA, and it is responsible for transferring or carrying the amino acid. So these are the major RNAs that we needed to know. Does anyone have any doubts in anything up until now?
No, ma'am.
Okay. Now moving forward, talking about transcription. Now see, the process through which the DNA turns into RNA or mRNA is known as transcription. Now, transcription process takes place in prokaryotes also, it takes place in eukaryotes also. So we have to do a comparative analysis when we understand this. Now, talking about the prokaryotic transcription. Now, in this one, remember that since prokaryotes do not have a very proper defined cell, right? Everyone remembers prokaryotic cell. We know that prokaryotic cell is not very organized. It is just there haphazardly. So the process of transcription takes place in the cytoplasm only because there is no other compartment. Now, for the process of transcription, they take the help of transcripting enzymes. Now, what are the enzymes? The enzymes that are like the transcripting enzymes that are present in the prokaryote, they are DNA dependent RNA polymerase. Why DNA dependent? Because from DNA only we are making the RNA. For the formation of RNA, we require polymerase. So the polymerase will be RNA polymerase, but it will be DNA dependent because we are using the DNA as a template. You people use template for drawing, right? Stencils. So imagine DNA as being the stencil over here. Okay. Now, see RNA polymerase, this RNA polymerase, it has two factors connected to it, or you can say associated with it. One of the factor is sigma factor and one of the factor is rho factor. Each of them has, you can say, their own functions to perform. See, the sigma factor is the initiation factor, meaning it causes the initiation or the starting of transcription. The rho factor is the termination factor, meaning it terminates the process of transcription. Okay. See, Friday, human reproduction, molecular. This is for another batch. Okay. Now, talking about the process of transcription and how it happens. Okay, now this particular enzyme, which is DNA, always remember students, whenever you're writing about transcription in the case of prokaryotic cell, remember that the transcription, it happens with, like you have to remember all of the enzyme names. Apart from that, remember, whenever you're writing about RNA polymerase, never just write RNA polymerase. Say that it is dependent on the DNA. So it is DNA dependent RNA polymerase. Now there are 3 steps to transcription. And remember that this RNA polymerase, it can transcribe all types of RNA, meaning it can make mRNA, it can make tRNA, it can make rRNA, it can make all types of RNA. Now, the first process is initiation. Now, in this process, the DNA-dependent RNA polymerase, it binds to the promoter area. You can say the gene, it has a promoter area. So, this comes and binds to the promoter area and initiate the transcription process. The starting of the process, meaning you can say the switch is on now. Okay. After that comes the elongation phase, meaning the RNA polymerases, they use nucleoside triphosphate as their substrate, and they start making complementary RNA. For example, okay, now I have such a nice question in my mind. Okay, see this. Okay, this is one sequence. Now, from this sequence, you people have to, you, from this sequence, you people have to tell me the sequence of its complementary DNA and the sequence of its complementary RNA. Tell me, what will the sequence be from this particular sequence? You people can note down and you can do, or you can say orally also. You can note down also and then tell me. Hmm. Tell me the answer, students. Now, what is first? Tell me about DNA. Hmm? Yes, tell me what happened.
I
Yes,
don't know how to do it.
you people know it. It's so easy. I mean, this is the easiest thing. You just have to make the base pairing. So, complementary to that. So, A pairs up with T pairs up with A, T pairs up with A, G pairs up with C pairs up with G. Meaning the ones that they are complementary to. You have to find out the sequence of this DNA. Meaning we are using this as a template and now making our own sequence. So we have done till here. Now A pairs up with T. Then C pairs up with G. This G pairs up with C. The T pairs up with A. The A pairs up with T. And lastly, the G pairs up with C. What was so difficult in this? This is the easiest question that I can ask you ever. Now tell me the same thing for RNA. Are you people getting it? Where are the rest of you? Switch on your videos. Come on, quickly. No one will switch off their videos now. Switch on your videos. You can't switch it off until I ask you to do so. Tell me the sequence of RNA. Just like that. Do it. It's just like that. Say Students, can you hear me? Please switch on your video, or else I'll start calling your parents. Please, don't make me do that. Okay, for RNA, the adenine pairs up with what? Tell me, adenine pairs up with?
Ma'am, guanine.
Hmm?
Guanine.
No, I just did it. Adenine pairs up with thymine, right?
Time. Oh,
But instead of thymine, what will be present in RNA? Uracil, because thymine is not present in RNA, right? You people are not getting this thing? Tell me yes or no. Say something so that I'll understand. Siddhika, are you understanding what I'm trying to say?
I didn't literally.
You're not understanding. Amna, what about you? Are you understanding?
Yes, ma'am.
Then tell me the answer. Tell me the answer. Tell me the rest of the sequence of RNA. Tell me, students. See, the thymine, if even if instead of thymine there is uracil, the uracil pairs up with adenine, meaning you have to make complementary base pairs. Just remember that adenine will always pair up with thymine and guanine will always pair up with cytosine. With this only, you have to make the complementary sequence. What is so tough in this? Adenine will pair up with thymine, but in RNA, there is no thymine. Instead of thymine, we have uracil, right? So now the uracil will be doing that work. Now adenine and now this thymine, instead of this uracil will be present, but it will pair up with adenine. We have to make the pairing. This is known as complementary base pairing. This is what happens during the process of transcription. You people still you did not get what I'm trying to say in this particular topic. You can note this down and it's okay if you're not getting it right now. Just dwell on it a little bit. We'll discuss it later on also. Now, let's talk about transcription first. So, talking about transcription, so it uses the template strand as the, you can say, substrate, and it starts adding complementary base pairs. Now, it polymerizes in a template dependent fashion, just like how I gave you a template and I asked you to make the result of it. Same way, the elongation process happens like that, and it follows the rule of complementarity. This is the rule of complementarity, meaning guanine pairing up with cytosine, adenine pairing up with uracil if it is RNA, and adenine pairing up with thymine if it is DNA. But thymine, DNA we're not making, we're making RNA. So the adenine here will pair up with the uracil. Is it clear? Now, what happens? The helix opens and the whole DNA gets transcribed, meaning transcription happens through the whole DNA. Lastly, there is the termination. So once the enzyme reaches the terminator area, the RNA falls off with the RNA polymerase. This results in the termination of transcription. So this is what happens. See, initiation, elongation, and termination are the three processes. The first is that promoter, which is sigma. It is coming and sigma factor is coming to the promoter and attaching to it, because of which initiation of the process is happening. Then it moves along like this throughout the entire length of the DNA and it starts adding, you can say, complementary base pairs. And lastly, this RNA falls off, and this is what the RNA that has been made. So transcription is completed. Now, in the case of prokaryotes, since they do not have compartmentalization in their cell, So what happens? That after the, like during the process of transcription is taking place, then only the translation also starts, which is protein formation. And remember that the transcription and the translation, both of these processes will be taking place in the cytoplasm or the cytosol. Clear? Now why? Can anyone tell me why? Again, all of your videos got switched off. I don't know. Why are you people in the rebellious stage? Yes, tell me why transcription and translation takes place in the same, or you can say in the cytoplasm. Because there is no compartmentalization. Okay. Students, I will not carry on the class if your videos are not switched on. I will not take the class further. I do not like this. Why this disobedience?
Can you
I
see
don't
me?
understand. Hmm?
Can you
Yeah,
see
yeah,
me?
yeah, you are there. I'm talking to the rest of them. You people have your video switched on and then you switch it off. Why? Is it some, like, is there something important going on? Tell me, Amna, is there something important going on in your side?
No, ma'am.
Then why are you switching off your video again and again? What about you, Ikra? Tell me. Now, you people, I'll like ask you separately. Tell me, Ikra, why? What is the reason for you switching off your video? If there is a reason, maybe I'll understand if it is a valid reason. What is the duration of your class, students? What is the time duration of this class? Tell me. Anyone can answer. Tell me what is the time duration of the class? 1.5 hours, right? 1.1 hour, 30 minutes, isn't it? Now you people can't keep your video switch on. You can't concentrate. You can't learn for 1 hour, 30 minutes. Is it so hard? Why is it so hard? Is it the chapter? Is it me? Tell me, what is the problem? Still, even after I am saying this much, Siddhika still has her video switched off. I don't understand this. This is not how I like. You know what, students? I'd hate to say this, but you know, like none of the boys are here who normally attend the classes, but they are better than you all. They at least listen to me. Why are you all behaving like this? I really don't understand. I don't want to be scolding you people all the time. Please don't make me do this. I am not this. I am the fun teacher who makes joke with you, who laughs with you. Please don't make me do this. I really hate this. If you can stay focused, if you can concentrate 1 hour, 30 minutes, like today, none of you have answered one question. Not even one question. None of you. All of you are sitting here blank. None of you. Even after, like, we are in the middle of the chapter, almost nearing to the end. And no one can answer one thing even if I ask. Who is given the double helical structure of DNA? Siddhika? Who is given the double helical structure of DNA? Amna, can you tell me? Who gave the double helical structure of DNA? What's the use? What's the use of teaching you then? How will you do it if this question gets asked in the test? What will you do? I am teaching, I'll say that I have completed the chapter. What you will do? Please be serious. You people are in the senior most class. Okay. All right, now moving forward, see the So termination has happened, meaning the RNA which has been formed, it comes out. Now remember that after the process of transcription, translation takes place. Now translation is the process in which protein formation takes place from RNA. RNA produces the protein with the help of certain sets of enzymes. But remember that transcription and translation, they take place in in one cytoplasm in the case of prokaryotes. Now, I asked you people why. The reason why is because prokaryotes do not have compartments in their cell. They just have normal cell. They have a very basic plan and they do not have any cell organelle. They do not have a nucleus. So that is why. Now, also many a times the process of translation or the process of translation can also be called as protein synthesis. It starts much before the mRNA is fully transcribed, meaning while the mRNA is coming out of here, then only the transcription of that part which is hanging, it starts. It doesn't even wait, since the whole both of the things, they happen in one compartment only, which is cytoplasm. So as soon as the RNA comes out, translation of it happens, meaning that RNA starts making the protein. Also, many a times, yeah. So this much is clear to everyone. Prokaryotic transcription is clear. What are the 3 steps that take place in prokaryotic transcription?
Initiation, elongation, and termination.
Right. Okay. Ikra, tell me the name of 2 factors that are there, the important factors that are responsible for transcription. What are the names of 2 factors? Yes, Siddhika, you tell me. What are the names of the 2 factors? See, if you people don't know, then say, ma'am, we don't know, so that I can ask someone else. Yes, Mariam, tell me.
Sigma and rho.
Sigma and rho. The other names for sigma and rho are
Initiation,
initiation factor and termination factor.
termination factor.
All right. Now talking about the process of transcription in the case of eukaryotes. But before that, you people can note down. And if you people are having any difficulty, please tell me so that I can clear your doubts, because you're not answering even like simplest of the questions. So if you have any difficulty, do tell me, okay? I'll try to help you. Okay, now you people can note down. Now for noting down, you can switch off your videos, but please switch it on when I am teaching you.
I'm done.
All of you are done?
Yes, ma'am. Done, ma'am. Done, ma'am.
I'm wondering.
You're writing? Anyone's left?
Yes, ma'am. Just a
Okay,
minute.
okay. Okay, okay.
Ma'am, done.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Done, ma'am.
All of you are done?
Oh, yes, ma'am. I'm done.
Okay, everyone's done?
Done, ma'am.
All right. Okay. Now moving forward, let us talk about the eukaryotic transcription and how it takes place in the case of eukaryotic organisms. Now, since there is a proper compartmentalization in the eukaryotic organism, so the transcription, it does not take place in the cytoplasm, rather it takes place in the nucleus. But in the case of eukaryotes, there are so many complexities because of compartmentalization, and since they are higher organisms. Now, the one of the major complexity in the case of transcription of eukaryotes is that there are RNA polymerase present in the nucleus, as well as these RNA polymerases are present in the organelles also, meaning more than needed RNA polymerases are present. Now, three RNA polymerases, they are present in the nucleus. The first one is RNA polymerase 1. They transcribe the following RNA, meaning with this RNA polymerase, we can get 28 sRNA, 18 sRNA, and 5.8 sRNA. These are the different versions of RNA. Then with RNA polymerase 3, the transcription of tRNA, 5sRNA, rRNA, and snRNA. SN stands for small nuclear. So these kinds of RNAs can be made or can be transcribed with the help of RNA polymerase 3. Then comes RNA polymerase 2. So with this RNA polymerase, we get the hnRNA, that is heterogeneous nuclear RNA. Now, this hnRNA is also known as the precursor of mRNA, meaning you can see here that none of the RNA polymerases transcribes mRNA. They are transcribing rRNA and they are transcribing tRNA. But there is no mRNA mentioned here. Instead of mRNA, hnRNA is formed, and then from hnRNA, mRNA is made. Clear? So these are the various kinds of, just like how in the prokaryotes, DNA-dependent RNA polymerase was present. That's it, right? But here, three different RNA polymerases are there. Now, after the process of transcription is completed, meaning initiation, elongation, and termination is done, then the heterogeneous nuclear RNA, it contains exons and introns. Introns makes the RNA non-functional. For example, if this is the hnRNA, so it will contain structures like this. These are the introns, and these are the exons, like that. So exons and introns are present on it, and intron, this is of no use. It makes the RNA non-functional. So the removal of intron is very necessary. So for that process, splicing is required. Splicing is the removal of introns. We cut it off, not we, the cell cuts it off, and the exons are then joined together. So this is one such modification after transcription. Now, two additional processes are required. See, these are the reasons because of which the transcription in the case of eukaryotic organism is very complex. Apart from this splicing, then additional processes are required, such as capping and tailing. Now, in the capping process, methyl guanosine triphosphate is added at the 5' end of hnRNA. Meaning, wherever is the 5' end, at that place, methyl guanosine triphosphate is added. And in the case of tailing, tailing means at the 3' end, 200 to 300 adenylate residues are added. Adenylate residues, 200 to 300. So this is known as tailing. So one thing happens at the 5' end and the other thing happens at the 3' end. So this is sort of like, imagine it being a modification. Now once all of these processes are done to the hnRNA, then the hnRNA becomes mRNA, messenger RNA. Okay, this is the most important RNA. Now what happens? The mRNA comes out of the nucleus for the process of translation, that is protein synthesis. Also, remember these two lines, they are written as such in your NCERT, that the presence of introns is reminiscent of antiquity. The process of splicing represents the dominance of RNA world. Just remember or learn these two lines as it is. Okay. Why it tells us about the dominance of RNA world? Because the RNA decides to remove the introns and keep the exons. So it is showing its dominance. So the additional complexities in the case of eukaryotes, see the basic process is same. Initiation, elongation, termination is same for eukaryotes also. But the only difference is that in case of prokaryotes, it takes place in the cytoplasm. In case of eukaryotes, it takes place inside the nucleus. Now, in the case of eukaryotes, there are 3 RNA polymerases that transcribes different RNAs. mRNA is not directly made. First, hnRNA is made, which is the precursor of mRNA. Now, when additional modification happens to the hnRNA, then an mRNA is formed. That is how we make the mRNA. Now, what happens in the additional things? Modification. First, the splicing happens. The introns are removed and the exons are attached, or you can say joined. Then what will happen is that capping and tailing is done. Capping is done at the 5' end. Methylene guanosine triphosphate is added, and tailing is done at the 3' end, which is the 200 to 300 adenylate residues are added. And now finally, the hnRNA becomes the mRNA. Clear? So transcription in the case of prokaryotes and transcription in the case of eukaryotes, is it clear to every single person in the class?
Yes.
All right. Okay. Now you people tell me if you have any doubts or anything you want to ask me, because we'll keep it till here only. I don't want to confuse you all with a lot of things at once. And in the next class, we'll be talking about the genetic code and its features, mutation and everything. Okay. So till now, does anyone have any doubts?
No, ma'am.
All right. Okay, then let's just keep it till here only for today's class, as maximum students have left the class. Only 3 students are there. So we'll keep it till here only. And in tomorrow's class, we'll continue with the features of genetic code. Okay.